Thanks for the kind introduction, and happy birthday to the organization, 30 years. It's been a hiatus of, I think, 14 years since I was last on the podium here, so it's a real pleasure to be here today and to speak to the topic. And the topic in hand is why we need to deliver large amounts of powder to the lungs and the concurrent challenges, so I've abbreviated that up there a little bit because I think most of us in the room are familiar with the need for high-dose powder delivery, and that's the sort of topic of today. The way I'm going to go about this is to give you a brief background to why I think this is important and essentially give you the need. Then give you some examples of the formulation approaches that support this activity, so the drug and then to some extent targeting. I'll talk a little bit more about devices and various devices, which you know we really are in need of at this point, and then conclude really with potential challenges in a summary statement. So just before I start this, I should say that where there are acknowledgements, I've tried to recognize people on the slides where the work was done rather than have an acknowledgement slide at the end in the interest of time. And then you'll also notice that I'm actually going to acknowledge certain products. I'm not here to try and promote those products. I just really want to use them as examples as I go through the various technologies that are available to us. So let me first start with the need. If you look at infectious disease around the world currently, it's very serious, and we have a burgeoning antibiotic resistance problem. So my particular area of interest, which I'll refer to as I go through this, is tuberculosis, where we have a significant current multi-drug resistant and extensively drug-resistant disease. You're probably all aware of the recent incidence of non-tuberculous mycobacteria drug-resistant disease. There has been for some time an interest in cystic fibrosis infection with Pseudomonas aeruginosa, and then of course there are other diseases or other infectious organisms that are related to cystic fibrosis. And then of course we have ventilator-associated pneumonia and other pneumonias that are around the world that are so significant that Bill Gates recognizes this as an area of interest for the Gates Foundation to support. So the specific antibiotic resistances then are in TB, in a variety of different non-tuberculous mycobacteria, and particularly abscessus is one that's becoming one of significance. We have MRSA on top of the others that I cited up there of cystic fibrosis infection, and then the ventilator pneumonia. So one thing I did not put up there, but I recognize is a serious issue, is fungal disease, and particularly aspergillosis, and you're probably aware of the work that's going on in that arena, and I would shout out for Bill Williams there, who's been doing a lot of work on vancomycin, but there are others too. Since I'm not talking about fungal infections today, I don't intend to put this in the forefront of my activities. So I have used this slide before, and I make no apologies for it, because I think this is essentially a description of the way that we go about looking at these things. So there are a variety of therapies that are available to us for infectious diseases. We need to take into account the biology of the disease, and then ultimately the technologies that we are trying to develop are essentially to achieve a specific outcome. And so having said that, today I'm not going to talk about the biologics, and I gather you're going to hear about that later on anyway, because the real issue that we're focused on is the ability to deliver drugs, and they're usually fairly low potency drugs. The way in which the host parameters affect our delivery of these drugs, and then the fact that ultimately for these low potency materials we're going to need to deliver high doses. And so the question is what technology is associated with that. So with tuberculosis, which I'm going to focus on initially, there are a range of different therapies under development. This lists pretty much all of them. But what I'd like to focus on this slide is the disease itself. So the way that you contract tuberculosis is that you inhale a very, very small number of colony-forming units, which are ultimately housed in the macrophage. And for those people who are susceptible to disease, and it's a very small proportion of the total, 10% or less actually contract the disease. And of that 10%, fewer still manifest disease, and only of the order of several million a year are actually challenged by fatality with the disease. But essentially what happens is the organism occupies the macrophage recruits T lymphocytes, you eventually form a granuloma. That granuloma can become caseous and ultimately become a tubercle. But in the meantime, over an extended period of time, you get the basalemic phase where the microorganisms are transferred to the circulation, and eventually they come back as a secondary infection where they're much more significant in their effect. And this is where you largely get the serious manifestations of pulmonary TB from. So today I'm going to focus initially at least on drugs, old ones and new ones. We've worked on combinations, but I won't talk about that for the time being. So let me just focus for a moment then on formulations. So of course, any powder that we're going to use, we have to be able to formulate so that it would be successful in delivery in high doses. The drug itself may be used in conjunction with additives. Particle engineering may be applied. And there may even be other approaches that are adopted. But I'm going to argue for a moment that things like blending are probably not relevant to the particular approach that we're going to take. So we're fortunate in a way that an observation was made approximately 20 years ago about the behavior of particles, something that was evident from first principles, but nevertheless became an important feature in selecting formulations for high-dose delivery. So, of course, here's your latex cast of the lung with the periphery removed so that you can see the airways. And this is sort of, in some ways, a branching network that's self-similar at all levels of scrutiny. And I just want to characterize what happens in an airway in terms of particle deposition. So initially, we have inertial impaction, which is, of course, due to the mass and velocity of the particle in the upper airways. If you were to traverse those airways to get to a bifurcation that's further into the lung, so more to the periphery, so we're talking about maybe less than 5 micron particles, 1 to 5 microns by inertial impaction, and around about 1 to 2 microns that has not deposited by impaction will be subject to sedimentation. And then finally you have diffusion of those particles that approach the mean free path of the gas, which is generally less than about half a micron. It's less than 1 micron technically because that's where slip correction kicks in, but half a micron is where you see a substantial variation because of the diffusive nature of those particles. And of course, we're not particularly interested in those particles that are subject to diffusion. We generally aim to that 1 to 5 micron size range where sedimentation and impaction would dictate deposition. So others have talked about this at great length. We've known it for probably 50 to 70 years that if we have particles that are in a median size range of 1 to 3 microns, they'll deposit predominantly in the periphery of the lung, and of course there's some distribution around that median. And if you take a stripped-down version of Stokes' law that essentially predicts behavior, it looks like this. So ultimately, the aerodynamic size is equivalent to the geometric size multiplied by the square root of density. This is for spherical particles that are greater than 1 micron in size, and this is for Reynolds numbers of less than 1. So there are a lot of assumptions have been made to make Stokes' law essentially collapse to this form. So nobody in the room is going to be surprised that this was the basis for the observation from Dave Edwards and others at MIT that regarding the use of large porous particles for pulmonary drug delivery. And the idea being that the aerodynamic size for very low density particles will mean that you have fairly large geometric sizes to give you a small aerodynamic size. And the original intent of that work was to avoid macrophage uptake. But an observation that I would credit Jeff Weers with, if he's anywhere in the audience, is that if you have large particles, low density particles, you've essentially moved the point charges such that now you have larger distances between point charges or points of mass in the particle, and consequently you've done the thing that everybody thought was impossible, which is you've reduced the van der Waals forces. You can't do that with solid particles. And also, by having a larger particle, you've made it subject to a greater interaction with the airflow, because now it projects into the airflow. And then, of course, because the forces holding these particles together are fairly low, any sort of impaction or turbulence will very readily disperse these particles, so you can get efficient delivery. And it was largely this observation that allowed us to go after high-dose aerosols. And of course, probably the best example is this, with credit to Jeff again and Dave Geller for this particular paper. But of course, a very simple device is needed. These sorts of particles will spontaneously generate under the convective forces of air in the room. So if you had them on a watch glass here, they would just spontaneously generate as aerosols, just due to their interactions. This is a particularly unique type of low porosity particle in that it's a so-called waffle ball, as opposed to collapsed hollow spheres that you may have seen, or hollow spheres that others have used and that appear in the literature quite frequently. And so I would argue that it was this observation about, and these are spray-dried particles that have low density, that allowed us to step beyond the classical milled particles blended with lactose, which are relatively inefficient at delivering drug, but since the sorts of drug you need for asthma and COPD are very, very low dose, you can live with that inefficiency to some level. So, of course, for this particular product now, we're talking about a dose that's delivered from 4 capsules of 112 milligrams. So you've gone from, I believe the largest product of an asthma product was from a dry powder inhaler was around 1 milligram, but we're frequently at the sort of hundreds of micrograms, if not individual micrograms of drug for asthma and COPD. And here we are now up in the 112 micrograms, so over 100 milligrams of drug. So very large doses of drug. Okay, so with that background then, so we have a technology that we can employ to actually deliver high doses of particles. And so we took an approach with pyrazinoic acid, which is the active moiety in pyrazinamide, one of the major drugs, first-line agents in the treatment of tuberculosis, spray-dried particles of this, characterized the particle size and had a certain fraction in the appropriate size range. And then we delivered that in addition to oral RIF. And so this allows me to make the first point that I would make about aerosols for TB. You will not treat TB with a single drug. The first line approach is four drugs. And for drug-resistant disease, you're often over 10 drugs. But aerosols, by their targeting and the fact that you can give fairly high doses locally, would be a supplement to oral doses, and obviously it would have the advantage of local delivery. And the key advantage that I think that it offers, I'll mention in a moment, but first let me talk about what happens when you use the aerosol. So you have untreated, these are guinea pig lungs, by the way, that we're looking at sections of, untreated animals, those that received oral RIF, those that received oral rifampicin, I should say, and oral PZA, so this is part of the standard of care. And these were at doses that are relevant to humans. And then RIF, and then an aerosol of pyrazinoic acid, the active moiety. And I don't really have to point out to you what's going on here. These are granulomas, some of which are necrotic, and you pretty much have a few immune cells in the lungs of the aerosol-treated animals, but otherwise they're quite healthy. But the key point that I'd like to make about this, because I'm not going to dwell too much on TB as I go forward, is that when you look at these animals, a significant reduction in the host pathology in response to the treatment occurs, and notably with so-called type 4 granulomas, which we think are primaries. And this we basically assert this based on consultation with people that work with these animal models. And then this appeared to be preferentially affected to type 1 granulomas, which are secondaries. The primaries are the ones that would be those that were formed when you initially inhale and get infected with the microorganism. And the secondaries would be those that come from the vascular bed. So the dogma is that with persistent disease, you actually see it in secondaries first. But I would argue that with persistent disease, which basically is very hard to find, it starts in primaries in very small numbers that you could never plate, and then it spreads and ultimately shows up as secondaries. What this means, the fact that we can target these primaries, is that we actually have an approach to treating persistence. And if you wanted to take the long view in terms of eradication, we might even have a treatment for latency, but that's a much harder task. If we can treat persistence, however, we can sterilize people and ultimately prevent them from having disease going forward. So we've published this. This is a collaborative work with Miriam Braunstein. So having said that then, and pyrazinoic acid, by the way, is an old drug that appears to be useful in an inhaled form. CPZ45 is a new drug. It was discovered by the Institute for Microbial Chemistry in Japan. And I work on this in a collaboration with a number of different organizations shown there. It's active on a new target, which is a cell wall core, so WEC-A synthesis, which is known as WEC-A. So the WEC-A is the target. This drug will work against MDR and XDR TB, highly water-soluble, but has low oral availability. So consequently, to achieve high lung concentrations and minimize toxicity, we believe that dry powder approach is valid. And we've conducted some work on this through funding from NIAID. And a lot of the animal work was conducted by different groups, notably by Lucila Garcia-Contreras. So we prepared particles that are shown up here. These are more of the collapsed hollow sphere type particles. They're amorphous when you characterize them. We've been doing this now for over 5 years through a spray drying process. And we can make them in sort of the gram quantity scale using a small spray dryer, the Buki B290. They're very stable, and there is no excipient associated with these drugs. In fact, this drug will stabilize other drugs. It just happens to be a feature of the drug. So the consequence of that is that we can deliver 100% drug, and there's no dilution with any additives. I won't dwell on the cascade impaction, the X-ray diffraction, and then the sort of confirmatory laser diffraction for aggregation and so on. But we've done a whole host of tests on the physicochemical characteristics of these powders with 18-month storage at various relative humidities, and including in desiccant pouches. There is an accelerated storage, so at 40 degrees we do start to see some moisture uptake. So where we are with this project now is that we're essentially going from the efficacy work, which we've concluded, into the preclinical studies. And so the work that we'd concluded with the funding that I mentioned a moment ago was in PK and healthy animals efficacy studies in infected animals using spray-dried doses that are equivalent to 2 milligrams per kilogram. We see a significant reduction in the burden of bacteria. This is a monotherapy. We only do this because in order to see a substantial effect, you need to not have other drugs there. You can imagine if you have 2 or 3 of the first-line agents in, you've already knocked it down so that you may see a very limited effect of an added drug. But that doesn't mean that we would ever use this on its own. We see the efficacy is 5 times higher than the equivalent subcutaneous dose. We've completed in vitro studies under the auspices of Lilly, one of the partners, in the classical assays to do with PK and toxicity. We've conducted both rat and dog studies at Lovelace Respiratory Research Institute with Phil Keel, who's probably out in the audience there somewhere, at a variety of different doses, and the drug was tolerated very well. So we are happy that we're moving in the right direction in terms of moving this drug towards the clinic. We're currently funded to actually do development work with this project, and we spent most of our time in this first period. We were funded for this work starting earlier this year to do the scale up from the small scale spray drying to the larger scale spray drying. But along the way, the other piece of this that we've been filling in with is building our documents for the IND. We won't, of course, get to the rat and dog toxicology that's shown in the middle there until we actually have the process to make this cGMP material. We've been, until this point, making non-GMP material. So we've gone into the larger scale spray drying. So this is a mobile mini. I'm sure many of you have seen this. The particles are looking fairly similar. This is just a more lucid particle than the image, rather, than the one I showed you earlier. We're looking at a variety of different ways to collect to sort of increase yield on these, the method. But if you look at the median sizes, which are in that sort of 2.5 to 3 range, we're fairly happy with the way this is going so far, and the GSDs are around 1.8, 1.9, which is what I would expect for a spray-dried formulation like this. And this product will ultimately end up in pre-IND early or spring of next year, and then hopefully we'll be at IND by 2023. We have a lot of CMC work that needs to be done under GMP conditions or GLP conditions to support the IND submission. So the program is going very well. It's basically in preclinical, formal preclinical talks. Many of you have heard me talk about capreomycin, which is another drug that we did many years ago, which went through phase 1. And we have a combination of capreomycin and CPZen, which is through rat GMP tox, and hopefully that will come in quite close behind. So, anyway, for high-dose delivery of these spray-dried particles for the treatment of TB, there's a lot of work going on. I'm not the only one that's doing it. I'm just sharing my data, and credit to all of you who are doing work to try and treat TB. It's an urgent medical need around the world. So the thing about TB is, as I've already said, that you will necessarily be taking some form of dose for the systemic manifestations of disease, usually a tablet. And the idea is that we can supplement with an aerosol, and therefore we can penetrate into regions of the lung at high doses that might not ordinarily see the drug. And by the way, for many years I speculated that part of the reason for persistence of TB disease was that, contrary to people's beliefs, oral drug delivery simply did not get the drug to the lungs in sufficient quantities or penetrate into granulomas in sufficient concentrations to treat the disease. And there's evidence for that in the literature, and I would guide you to Veronique Dartois' work on the differences that you see in drug penetration into granulomas and how that could give rise to resistance. And of course, by giving an aerosol like this, we completely obliterate the possibility that you'll never get the drug. The drug is right there, and it's going to drive, since it's at a high concentration, it's going to drive into the granuloma in a way that an oral dose couldn't do. But you actually have a better situation when it comes to non-tuberculous mycobacteria, which I'm sure is also an interest of some of you in the audience. We're not now concerned too much about systemic elements of the disease because it's largely a pulmonary disease. And so what we really want to do is simply use the aerosol and get it to the site of action to treat those organisms in the lung. And so I think people see this as a very logical approach. There's some equivocation about aerosols for TB. There doesn't seem to be any equivocation about using aerosols to treat NTMs. And of course, it's a topic of conversation, and Thomas Hoffman, who has an interest in developing products for this purpose, has published on the subject if you're interested. And of course, quite recently, we've had this product, the amikacin liposomal formulation, which is delivered in a very high dose for a given period of time. Sorry, this should be minutes, sorry. You know, we have a model in a way of giving very high doses for the treatment of NTMs, and this, of course, is mostly for MAC, Mycobacterium avium. So can we give high doses of a drug to treat NTMs in a dry powder device? Well, one of the things that I draw your attention to, so it just allows me to make a point, is when you're using these very low density particles, then necessarily quite large volumes are required to accommodate the mass. I mean, the individual particles hold very little mass, and consequently you need a lot of volume, and hence, by the way, the 4 capsules that you saw for the tobramycin product. So is there a way that basically we can use a slightly smaller contained volume by matching it with a formulation mass under conditions that would allow you to deliver it? Again, Thomas Hoffman has been doing some work on clofazimine, and basically he's doing this with nebulized therapy, and as I understand, it's moving very quickly. Clofazimine has the unique property of essentially penetrating into macrophages and targeting the macrophage through the activity of the drug rather than any specific approach. And Hugh Smythe has taken an approach where he's using these milled particles that aggregate into essentially, if you think of it, porous aggregates. And for any of you that attended the ISAM meeting in 2017, you will have heard this before. Jeff Weirs, again, has been espousing this, that there may not be a need to go to these ultra-low density particles, but actually what you could do is create particles that are essentially low density to some degree, but are made up of solid, smaller particles. And this is what Hugh is attempting to do here. And what this would do is it would allow you to put a larger mass into a smaller volume, and presumably you would need a smaller number of metering units, and maybe this would be useful in terms of patient compliance with the therapy. In terms of devices themselves, so there are obviously a number of different devices. I am, as many others, have been using the cyclohaler for a while, and had used it, by the way, for using the capryomycin study. We were aware of the Groningen system from Eric Freilink and Anna de Boer, and this would seem like an admirable solution to the not having to put blisters into capsules into a device, but rather just having a single foil in a disposable device. And both of these devices are being developed towards sort of larger dose delivery. So currently they're at the level of maybe tens of milligrams, and ideally you would like them to be in the 50 or more milligrams in a single dose. And so you have large capsule development by Plastiapi, and you have the Cyclops, which is a large dose device from the folks in Groningen. And the Groningen team have been fantastic at essentially looking at the performance of their devices, looking at specific drugs for TB, particularly isoniazid. And they actually do have and have published a single case study in which they treated a patient with colistin as a way of managing an extensively drug-resistant disorder, and done that effectively. So we have at least one case study in which this approach has worked. By the way, we also have 50 or more years ago, when streptomycin resistance was prevalent, many examples of the use of inhaled streptomycin in clinical setting, in off-label use by doctors who are at their wits' end trying to treat TB. So it's not as if we don't have evidence that aerosols work, they do. We just need products that can support the therapy. And then there are some other high-dose devices, and the orbital is one that you may be aware of from Pharmaxis, originally used to deliver mannitol. And I'd like to recognize Paul Young and Danny Trainee, who've done a lot of work on this device over the years. And then Drew Jones, who's at Manta, basically is working on a number of high-dose devices. And I only mention these because I want to show you a little bit of data in a moment that uses one of the Manta devices. So Gary Phillips was kind enough to give me some more detailed images of the orbital in case you haven't seen it before. And it allows me to focus on one element of the metering system that I think is very important for all of these devices. So as you know, this device has a puck, which I'll show you in a moment how this acts. But if I could draw your attention also to the fact that with capsules essentially oscillate and vibrate as you inhale past them passively. And so there's an element of mechanical disruption that goes on with capsules, in the same way that you'll see with this, that there's a sort of mechanical impaction, if you want to think of it that way, on the wall that gives rise to dispersion. You see puffs of the aerosol coming from the edge of this system as it's operating. I have to wait because I'm not sure it'll go on to the next slide. Good. So I think many of these simple passive devices use this sort of oscillatory, vibratory mechanism to help disperse the powder into the inspiratory flow. So we did a couple of studies, and the next few slides I want to show you, it's sort of presented rather like a comparison, but I really don't want you or I don't want to suggest that I'm promoting one device over another. I've worked for a very long time with Plastiapi devices. They work very, very well. And I just want to show you that we can do something with high doses that perhaps you can already do with the Orbital, and may be possible in the future from other devices. And so we took the pyrazinoic acid powder formulation that I mentioned earlier in the presentation, did some NGI work on that, and we looked at the RS01, so-called cyclohaler, with a 10 milligram load, and then we actuated 5 10 milligram doses. You know, at 50 milligrams was sort of a somewhat arbitrary number. I was thinking that if you were going to hypothetically come up with a high dose, probably your best opportunity would be 2 50 milligram doses at around 100 milligrams, or maybe 3. We're probably not going to deliver much more than that in the short number of breaths. And I know that the RS01 can actually manage, a number 3 capsule can manage more than 10 milligrams. So again, this is just a study designed to illustrate a point. And of course, the particle size distribution doesn't vary when you go from the 1 dose to 5 doses collected by cascade impaction. And so these are the comparisons we're doing. And then using the MANTA system, which was a 150 milligram dose, we can look at emitted dose, fine particle fraction as a function of nominal dose, or emitted doses. And I wouldn't, you know, again, to go back, I'm not trying to make any points about any differences you see here. The point is that you can get up to high doses by combinations of individual low doses, if you like, to get your high dose. Or in an ideal world, ultimately, we'll be able to get to a single high dose. And the reason for this is to ensure that we have patient compliance, which may be difficult otherwise. I'll come to a point at the end, though, that is worthy of consideration when you think about these two different approaches to things. Okay, so not to spend too much time on this, but of course, when you're looking at these inhalers, you have to ultimately go through your from feasibility clinical batch supply production scale. And the real point I want to make here, and with a big thank you to the manufacturers that are in the room, is that you really need help from the device manufacturers going forward. I mean, that's an obvious thing to say in light of regulatory authorities, but I think at a personal level, they make your life so much easier, and I'm really grateful for the help we've had so far. I can't finish this whole notion of devices without saying that, of course, there is a way that you can deliver high powder doses. That's almost by a so-called powder nebulizer approach. And I should, full disclosure, tell you that I'm on the board of CARE Biotherapeutics, which is run by Donovan Yates, who's a founder. And this is a powder generator that works on a basis of condensation, spray, evaporation, and then collection, if you like, of particles in a smaller volume of air. And you can generate particles of different sizes based on the gas that you're actually using. The efficiency is very high in each of these, for each of these different systems. And the delivery rate, if you look, this is on milligrams per second, you can tell that you can get up to 100 milligrams or more fairly rapidly. And of course, I mentioned earlier on that we have problems with ventilator-associated pneumonia, and there's a situation where you're not really going to use a handheld powder dispersion system, and this might be ideal. And moreover, if you have a particularly tricky manifestation of some of these other diseases, and you need even higher doses, this may be a strategy that goes beyond a nebulizer and gives you that high-dose rapid delivery. Okay, so I'm coming into the home stretch here a little bit. So what I've talked about to this point is drugs and basically the need for high doses and how that interacts with or is delivered through the patient. But you could take another strategy, which is to target the disease where it is in the host. And then it's a manifestation of the host immune cells and immune system, the pathogen, and your ability to target. And so I want to shift then for a moment to microparticles and nanoparticles, which is, if you look at the literature, that's probably been the dominant theme for those folks that are interested in treating TB to date. And again, for those of you, actually for those of you that were here the last time I spoke, you may have seen this last couple of slides. They've certainly been around a while. And they just allow me to make the point that you can manufacture particles. These are PLGA particles that contain rifampicin. They will penetrate into macrophages and be retained for several days. This is a rhodamine dye that you can see several days out after these have been given to guinea pigs by aerosol. And if you look at the treatment, you actually can treat both the lung and the spleen, so you're preventing the spread of disease. In comparison with rifampicin treatment alone, PLGA particles alone are untreated. So this is therapeutic, it's very targeted, and this is in the order of, I believe it was 40 microgram doses to these animals, which would translate into about, I don't know, about 7 milligrams probably total to treat the disease. And of course, if you want to deliver nanoparticles to achieve the same effect, you can put those in spray dried particles, and this is work from, originally Nick Zappas basically developed these so-called Trojan particles, microparticles that are made out of nanoparticles. These contain rifampicin again, and we collaborated with Dave Edwards, and the lead was Gene Sung, that actually did work on these sorts of particles. So nanoparticles, microparticles can be used to target macrophages. So in the last few minutes then, I want to talk a little bit about things we want to bear in mind. I don't know whether they're really challenges. They're more, for those of us that live with formulation device development, we're used to the idea that nothing's perfect from the beginning. But they're things that we need to bear in mind as we're developing these systems. So let's first of all think about what are the factors affecting target product profile. Well, the total dose is going to be an important thing. What is the potency of the drug against MIC? Most of these drugs are not particularly potent. And if you look at oral doses, they're being given in 600 milligrams to 1.2 grams a day. And actually, this is the big problem. If you have MDR-TV, you might be having 10 drugs at that dosage level. You're essentially having a breakfast cereal bowl full of tablets, and you're losing your entire GI tract epithelium, and you may die from malnutrition before you actually treat the disease. So we have to cope with that too. The doses to the lung will not be that high, but they're still fairly high. And then there's the feasibility of whether anybody can inhale the amount of drug that we want them to inhale. So, powder necessarily can give rise to cough, depending on whether you can avoid the cough receptors. But if you have enough powder going in there, eventually you're going to reach some sort of limit. And usually, by the way, in tox studies, it's the feasibility of delivering the powder that's the issue, not necessarily the drug toxicity. Do you need excipients? Well, if you have to add excipients, the problem is that that's going to increase the total powder dose. So if you have the targeting that I talked about a minute ago, where you had, I think I said I used 40 micrograms, the problem was that was in a 10 milligram dose with all the excipients added. So you're not really losing or gaining much, rather, in terms of the high dose delivery issue. What's the dosing regimen? How frequently do we have to deliver it? There may be an opportunity there. If we could dose more than once a day, we might be able to effectively come up with a therapy. And then what are you going to do in terms of therapy? Is it to prevent transmission? What sort of dose do you need for that? In addition to the standard of care, or are you creating a new regimen? The last is very unlikely, by the way. We've seen one new regimen in probably the last 30 years of oral doses. Again, coming back to this targeted delivery, in the end, what you really have is a combination of targeted, but you still need high dose. It's really hard to get away from that high dose need. All right, then quickly, in the concluding few minutes, What other things might you need to think about that have to do either with the device performance or with the biology? So here is an example of some work that I did years ago on it was actually on lactose products, but it allows me to make the point. So you have an impactor in which you can particle size the aerosol, and then just imagine you've got a dry powder inhaler there. This is just a standardized tube that we use. But then you can image the particles, or the bolus, if you like, of the powder as it enters the impactor. And if you do that, what happens is you get obscuration. So the particles enter the viewing mode, they slowly obscure until they maximally obscure, and then they exit, and you see limited obscuration. That sort of manifests itself, as this diagram suggests, is if you look at obscuration, your obscuration increases initially, maximizes as the biggest portion of the bolus goes through, and then it descends when you are clearing the viewing area. So if you go back to this classical paper from Clark and Hollingsworth, I don't know if Anne is here today, I just want to pick on this particular image and suggest that we're interested in this portion of the curve. So this is a deep breath, if you like, a maximum effort, which is what you might encourage somebody to do with a dry powder inhaler. This is the leading portion, the portion on which you'd like to get your aerosol if you wanted to penetrate into the lung. So I've sort of cut that here, and apologies for any this image if it's not too clear, but let me just show you what I think would be ideal. So in terms of the drug mass, is that the drug mass is distributed on the inspiratory flow fairly close to the beginning of the inspiratory flow. And I've if not instantaneous, close enough that you have expectation that we'll get to the periphery. I wonder, and I would defer here to those people that are working on ultra-high dose inhalers, whether that simply the mass itself will resist so that you get essentially an extended duration of release, which is simply because it takes time to disperse that amount of powder. And I think if you could talk to the folks who are working on high-dose inhalers, they probably figured this out. But I would point to the fact that this is something we need to be careful about, because anything that we see on the back end of this may not go as deeply into the lungs as you intend, and consequently would not be available to treat disease. Assuming this powder lands, we're used to, of the order of micrograms, hundreds of micrograms of doses, of drugs rather, to treat COPD and asthma. And we debate backwards and forwards how much dissolution matters there. Well, imagine now we're putting in 100, or maybe even 200 if we get really good at this, milligrams of powder. We've got at least two regions, the periphery of the lung, where you essentially just have surfactant on these fairly two-dimensional peripheral alveolar epithelial cells. And then you go up to the upper airways, obviously smaller surface area here, but you have columnar epithelial cells with cilia and mucus and so on. The medium for dissolution is different in both of these regions. And of course, there's a lot of stuff that goes in between. We have differences that exist, molecular composition of the various surface fluids, thickness of the layers, fluid volume, surface area. And so there might be some expectation that you would run into dissolution problems simply through saturation. So you would need to look at this in your PK studies and make sure that you weren't essentially accumulating drug. So in conclusion then, the incidence of lung infection and emerging threat of drug-resistant disease poses a challenge for antibiotic development, and therefore we need high-dose delivery drugs. And I think this is what's driving high-dose delivery at the moment. The advent of low-density particles introduced an opportunity for this, but I think what we're seeing is that we may be able to retain some of that low-density property without being ultra-low density, and therefore make it a little bit simpler from both a patient compliance perspective and also from a device development perspective. We can target intracellular pathogens, but then we won't gain much in terms of high doses. We'll still need high doses. We need to look at the intrinsic drug properties and what are the requirements for targeting strategies to overcome biological barriers. I didn't talk about that much, but for NTMs, for example, we may need to look at so-called biofilm busters to actually get us to where the organisms are. We need to look at performance of large dose powders, essentially scrutinize these things during development to ensure that it has desired dispersion and disposition properties, which is to say that when I bring those issues up at the end as challenges, I'm absolutely convinced that people are aware of this and they're working on it. But if you're coming into this new, be careful to think about it. And then the last thing, which I didn't mention, but I don't think it's worthy of a lot of discussion today, is the overall cost, ease of use, and the broad therapeutic and geographical relevance. Keep it simple, really. Keep it inexpensive. I mean, many of these diseases are found in parts of the world that can't really afford the sorts of inhalers we use for asthma. And I'm looking at 10 seconds, and I know we have questions. I think that's the last thing. Thank you for your time and attention. I'll be happy to answer any questions.